Today, we are going to discuss about periodontal microbiology. The topic will be covered under the following headings. So typically, 500 different types of species are capable of colonizing the mouth. Any individual may typically harbor 150 or more different types of species. This count in subgingival site ranges in healthy individuals from 10 to the power 3 to deep periodontal pocket 10 to the power 8. In general, these organisms live in harmony with the host, but in certain circumstances, a selected group of organisms have the potential to cause over disease, including dental caries and periodontal diseases. So, periodontal diseases are a type of infection. Destructive periodontal disease may be thought of as a series of infections that affect individual or multiple periodontal sites within the oral cavity. Evidence has accumulated that a selected group of species appear to cause these diseases, suggesting that the most appropriate control of these diseases is through the control of microorganisms that cause them. Coming to a brief history, from the era of 1880 to 1920, which is known as Golden Age of Microbiology, amoeba was discovered. It was found in higher proportions in destructive periodontal disease. It was also found in no disease site, not found in all sites of destructive disease. Local therapies were used, like use of dyes or other antiseptic agents, and amitin was used to eradicate them. Spirochetes, it was found in higher proportion in destructive disease. Therapies were proposed to control disease by elimination or suppression of microorganisms. Kritinsky and Seguin has done a systematic administration of neosalverin for controlling the disease. Fusiforms, as done by research, Plot and Vincent, have found these in destructive periodontal diseases. It was first related to periodontal disease by Plot, who termed them as Miller's bacillus. Streptococci, intramuscular injection of mercury or others, and it was used for vaccines. There were vaccines which were developed in first to third decade of 20th century. Three types were employed for control of disease, which are prepared from pure culture of streptococci and oral microorganisms. Then there were autogenous vaccines, which was prepared from dental plaque of patient with destructive periodontal diseases, stock vaccines as Van Cott's vaccines, and Goldenberg's vaccines or Inava endococcus vaccines. So these vaccines were administered systemically or locally in the periodontal tissues. In early years, bacteria was considered important in the pathogenesis of periodontal disease in early 1900. Beckwith et al. In 1925 had done a study in which streptococci and diphtheria within the epithelium were found also in underlying tissues. Gram-negative rods were observed in connective tissue. There was also a rare occurrence of spiral form in the tissues. Further, there was a decline in the interest of microorganisms. Belding in 1936 has said that bacteria are only secondary invaders and they were considered as dental orphans. It was thought that the periodontal disease was due to some constitutional defect on the part of patient, trauma from occlusion to disease atrophy or to some combination of these factors. By end of 1930, no researcher was actually working on bacteria as an etiologic agent. Coming to non-specific plaque hypothesis. So in 1950, periodontal disease was found that it was not controlled by treatment based on the constitutional defect or trauma from occlusion. For satisfactory treatment, plaque control was important, and specific type of species of microorganism were not important. So this was plaque hypothesis, non-specific plaque hypothesis, which says that bacteria are essential for causing the periodontal disease and removal of these bacteria is essential for the successful treatment. But which type of bacteria was not important. So accumulation of microorganism at a below margin below the gingival margin, leading to irritants, which leads to inflammation and destruction of periodontal tissue. This was the entire non-specific plaque hypothesis. In mid-1900, periodontal disease were believed to result from an accumulation of plaque over time, in conjunction with a diminished host response and increased host susceptibility with age. Several observations contraindicated the conclusion of non-specific plaque hypothesis. Some individuals with considerable amount of plaque and calculus as well as gingivitis never developed gingival-destructive periodontal disease. Also, site specificity came into picture. Return to specificity in microbial etiology of periodontal disease. In 1960, specific microbial etiology was resurfaced. It was based on two experiments. The first one, which demonstrated that periodontal disease could be transmitted in the hamster from animal with periodontal disease to animals without periodontal disease. And second, which demonstrated that spirochete was present in lesions of anal. By end of 1960, dental plaque was in some way associated. Low et al. And Thalid et al. Demonstrated that plaque accumulation directly preceded and initiated gingivitis. It was believed at this time that presence of bacterial plaque initiated a series of undefined events that led to destruction of the periodontium, and composition of plaque was thought to be relatively similar from patient to patient, from site to site within patient. Newman et al. And Slot demonstrated that microbial composition of subgingival plaque taken from disease site was different from the healthy site. Tanner and Slot have demonstrated that microbiotas recovered from chronic periodontitis different from the microbiota from healthy site. So this leads to specific plaque hypothesis, which was originated in early 1960, that certain plaque is pathogenic and its pathogenicity depends on the presence of or increase in specific microorganisms. This concept predicts that plaque harboring specific bacterial pathogens result in a periodontal disease because these organisms produce substances that mediate the destruction of host tissues. Coming to evidence of bacterial etiology of periodontal disease, it can be summarized under following heading: clinical observation, research done in last 250 years, and acute periodontal infection in which acute phase can be alleviated by antimicrobials. Relation of plaque level to gingivitis and periodontitis. So positive correlation between the amount of bacterial plaque and severity of gingivitis and amount of bone loss. Low et al. Demonstrated that intensive plaque control can eradicate clinical gingivitis. Clinical gingivitis can also be controlled by antibiotic or antiseptic agents. Linde et al. Demonstrated that the progress of destructive periodontitis could be halted and partially reversed by surgical procedures accompanied by twice monthly professional cleaning. Efficacy of antibiotic in treatment of periodontitis improved the therapeutic outcome. Post-immune response is seen and contribution of mechanical irritants. So pathogenic potential of plaque is seen in extraoral sites. Studies in experimental animals in rodent antibiotic or antiseptic can control infection. Disease can be transmitted from one animal to another. Introduction of plaque isolate developed pathology in germ-free animal. In dogs and primate, daily removal of plaque prevent gingivitis and periodontitis, and by immunization by organism or their constituent diminish the severity of periodontal disease. Criteria for defining periodontal pathogen. For more than a century, the classical Coates postulate have been used to define a causal relation between an infectious agent and a disease. The discrimination of a pathogen from a non-pathogenic species is not based on a single criteria, but rather on a weight of evidence evaluation. So Coates postulates were must be routinely isolated from diseased individual, must be grown in pure culture in laboratory, must produce a similar disease when inoculated into susceptible laboratory animals, and it must be recovered from lesions in a diseased laboratory animal. Sigmund Sokransky modified these criteria as it should be associated with disease as evident in number of organisms at disease site, which is an association. It should be eliminated or decreased in sites that demonstrate clinical resolution of disease with treatment, which is elimination. Demonstrate a host response in the form of an alteration in the host cellular or humoral immune response, that is host response. Demonstrate virulence factor responsible for enabling the microorganism to cause destruction of the periodontal tissues, virulence factors, and be capable of causing disease in experimental animal models, animal studies. Periodontal pathogen. The World Workshop in Periodontology, which is a consensus report in 1996, designated A. actinomycetum comitans, P. gingivalis, and T. forsythis as the most common periodontal pathogen, which are officially designated as periodontal pathogen. The prerequisites for periodontal disease initiation and progression includes infectious disease may colonize a host Host may not manifest clinical features of their disease for a long period of time. Disease progression is dependent on the simultaneous occurrence of a number of factors. The host must be susceptible both systemically and locally. The local environment has to contain bacterial species which enhances the infection. The pathogen must achieve sufficient number to initiate disease. The virulent periodontal pathogen. For a periodontal pathogen to initiate disease, three factors first be considered. First, the particular strain of virulent strain of pathogen should be present. Organism should possess all the genetic element and it should be present in right location in a site. Virulent pathogen, suspected periodontal pathogen in a periodontal healthy mouth or healthy sites in periodontally diseased mouth. So all clonal type of pathogenic species are not virulent. For many medically important pathogenic species, a very small proportion of clonal type account for majority of diseases. Example, virulent JP2 clone of AA in LAP. Certain clonal type of P. gingivalis are detected more often. Amano et al. In 2000 examined P. gingivalis FIM-A genotype in plaque sample from 380 periodontally healthy adults and 139 periodontitis patients. Type 1 and type 5 genotypes are most common in P. gingivalis positive periodontally healthy adults, while type 2 and type 4 in adults with periodontitis. Results suggested that the FIM-A genotype may be an important factor influencing the pathogenicity. Another important requirement for a pathogen to express virulence is that the organism should possess all the necessary genetic traits. The pathogen must be present in right location in a site, example in apical areas of the pocket or adjacent to the epithelium, and also they should be present in sufficient number to initiate the disease. Coming to the local environment, microbial interactions can be either beneficial to the host or they may lead to harmful mixed infections. The host compatible species could colonize sites that otherwise could be colonized by pathogens. These are some studies. Coming to host susceptibility, difference in disease pattern or severity may be difference in host susceptibility. Such factors include defects in PMN, poorly regulated immunological response, smoking, diet, and various systemic diseases. So these are few of these studies which shows how host susceptibility affect the pattern of the disease. Coming to microbiological aspect of host microbial interaction. For a periodontal pathogen to cause disease, it is essential that the pathogen must be able to colonize the subgingival area and produce factors that either directly damage the host tissue or lead to the host tissue damaging itself. Bacterial adherence is done with the help of certain surfaces, certain aspects which are present on the bacterial surface, which help in attaching of these bacteria to the tooth surface. Adhesins or receptors may be present. So adhesins are fimbriae or cell-associated protein which are present on the bacterial cell wall, and receptors are galactosyl residues, sialic acid residues, proline-rich protein, and type 1 and type 4 collagen which are present either on the epithelium or on the root surface. These are the examples of bacterial adhesins and target substrates present over the tooth tissue surfaces. Coming to a quality of bacteria which is known as co-aggregation, which means bacteria attachment or bacteria is attached to each other or the pre-existing plaque by different bacterial strain, which is known as co-aggregation property. Interbacterial relationship also play an important role in species survival. Some are favorable in as one species provide growth factors or facilitate attachment of the others. Other relationships are antagonistic. Bacterial evasion of host defense mechanism. So to survive in periodontal environment, bacteria must neutralize or evade the host mechanism. So the ability to adhere allow bacteria to avoid displacement by the host secretion. Desquamation of epithelial cell presents a new cleaning mechanism which is overcome by certain species by binding to underlying epithelial cells. Specific antibodies prevent bacterial attachment evaded by certain proteases changing surface antigen or mimicking the host antigen. They produce substrate which can suppress the activity of a or kill a PMN and lymphocytes. AA produce leukotoxins and cytolethal distending toxins. T forsythia and F nucleatum induce apoptosis. P. gingivalis inhibit production of interleukin 8. These all develop resistance against antiseptics and antibiotics. So these are selected bacterial properties which are involved in evasion of host defense mechanism. Factors that cause tissue damage include substance that damage the tissue cells like hydrogen sulfide, ammonia peptide, substance that causes cell to release biologically active substances like lipopolysaccharides, and substance that affect the intercellular matrix like collagenase and trypsin-like enzyme. So bacterial enzymes which are capable of degrading host tissues like collagenase are produced by P. gingivalis, A. actinomycetum. Trypsin-like enzyme is produced by P. gingivalis and AA and T. denticola. Keratinase is produced by P. gingivalis. So these are the examples. Also, these are the examples of the bacteria there and the effect of bacterial products on biologically active molecules. Like increased release of interleukin 1 is done by bacterial species AAF nucleatum and A. actinomycetum fomitans and P. gingivalis. The component involved is lipopolysaccharide and targets PMN. Also, they produce certain proteins like 37 kDa and LPS, which again targets macrophages and monocytes. Increased release of interleukin 6 is again done by AA and C. rectus E. coridens, produces protein like 37 kDa whole cells and they target the macrophages. Increased production of interleukin 8, TNF, and prostaglandin E2. Coming to health versus disease, microflora in dental plaque. So, this is a healthy microbial flora, and this, because of the potential pathogen, this will lead to transmission of this healthy microbial flora. Also, there are major ecological pressure leading to disease type of microbial flora. There is another hypothesis which is ecological plaque hypothesis, which says that plaque reduction will lead to gingival health and plaque accumulation will lead to gingivitis. Same way, increased inflammatory response will lead to gingivitis, decreased inflammatory response to lead to gingival health. So low GCF flow, predominantly gram-positive bacteria, all these will lead to gingival health, and predominantly negative bacteria, high GCF flow and increased inflammation will lead to gingivitis. So the bacteria which are destructive periodontal disease will only occur when there is susceptible host, presence of pathogen, and absence of beneficial species. Microorganisms which are present in periodontal health are streptococci, AA, gram-negative species like P. intermedia, F. nucleatum, capnocytophaga, Neisseria, and Vallona. Organisms which are associated with specific periodontal diseases. Chronic gingivitis, the plaque are mostly composed of Streptococcus sanguis, mitis, oralis, viscosus, A. neisselandii, P. micros. These are predominant gram-negative include Fusobacterium, Prevotella intermedia, Varicella parvula, Haemophilus, Capnocytophaga, and Campylobacter species. Specifically, pregnancy-associated gingivitis is seen in Prevotella intermedia. Chronic periodontitis cases, gram-positive will shift to gram-negative, cocci will shift to rods and spirochetes, non-motile to motile, facultative, obligate, anaerobes to fermenting and proteolytic. The predominant microorganisms seen in chronic periodontitis are these: AA, P. forsythis, F. nucleatum, P. micros, Prevotella intermedia, Prevotella porphyromonas gingivalis, Eubacterium. Predominantly active sites compared with inactive sites include Campylobacter rectus, Prevotella Porphyromonas gingivalis, Fusobacterium nucleatum, and Tannerella forsythia. In disease progression, Porphyromonas gingivalis, Prevotella intermedia, Tannerella forsythia, Campylobacter rectus, and Agribacter actimycetum comitans, and their elimination is improved clinical response. In recent studies, certain viral microorganisms are also seen. In localized aggressive periodontitis, predominant microorganisms are gram-negative capnophilic anaerobic rods, mainly harboring A. actinomycetum fomitans. Other microorganisms which are found are P. gingivalis, E. coridens, Campylobacter rectus, F. nucleatum, etc. In necrotizing ulcerative gingivitis and periodontitis, Prevotella intermedia, Fusobacterium, spirochetal microorganisms are seen. In advanced cases, spirochetes infiltrate. In abscess of periodontium, these microorganisms are seen. In periimplantitis, Actinomycetum comitans, P. gingivalis, T. forsythia, P. micros, along with some species like Pseudomonas aeruginosa, Enterobacteriaceae, Candida albicans, and Staphylococcus are also frequently detected. So aggregative actinomycetum formitans, which was formerly known as AA, it has one of the strongest association between a suspected pathogen and a destructive periodontal disease. It is a small, non-motile, gram-negative, saccharophilic, capnophilic, round-ended rod that forms small convex colonies with a star-shaped center. It was first isolated in 1912 and called as Bacterium actinomycetum formitans. In 1921, it was referred as Bacterium comitans. The genus name refers to a star-shaped inner structure sometimes seen in colonies. Now it is known as aggregative vector actinomycetum formitans. This is the summary of data suggesting its pathogenicity. It is most closely related to members pastoralissae. It is fastidious, facultative, anaerobic, non-motile, non-sporing, small gram-negative rod. Microscopically, the cell appears cocobacillary. 6 serotypes have been recognized A to F. Distribution of serotypes may vary according to person's geographical location and their ethnicity. Serotype B is commonly seen in US studies. C is seen in Japanese, Chinese, and Korean and Turkish, and A, B, C are seen in Finland, Sweden, and Denmark. Virulence factor. So it has many virulence factors like factors that promote colonization, factors that interfere with host defense, factors that destroy host tissue, and factors that inhibit host tissue. So these are the virulence factors of A. actinomycetum formitans. P. gingivalis. It is a second conscious periodontal pathogen. This organism is essentially absent during the periodontal health, but seen during disease progression and in established disease. These are non-motile, asaccharolytic, obligatory, anaerobic cocobacilli exhibiting smooth and raised colonies. When grown on a blood agar surface, the colonies are initially white to cream colored. With time, 4 to 8 days, these colonies darken from their edge towards the center and a deep red to black color which correlate with the concentration of proteodyne. These are the summary of data which suggest its pathogenicity. It can inhibit migration of PMN across an epithelial barrier. It is shown to affect the production or degradation of cytokines by mammalian cells. P. gingivalis Fim A gene encoding fimprelin. It is a subunit of Fimbriae. It has been classified into 5 genotypes, type 1 to type 5, based on their nucleotide sequences. Type 1 and type 4 genotype were most common in P. gingivalis positively healthy adults, while type 2 and type 4 were more common in subjects with periodontitis. So these are the virulence factors which are associated with the production of produced by P. gingivalis. Tannerella forsythia. It is the third consensuous periodontal pathogen. It is an anaerobic gram-negative member of cytophage bacteroids family and was initially named Bacteroides forsythias by Tanner et al. It was reclassified as Tannerella forsythia by Sakamoto et al. It is difficult to grow, often requiring 7 to 14 days for minute colonies to develop. It is gram-negative, anaerobic, spindle-shaped, highly pleomorphic rod. Growth was shown to be enhanced by co-cultivation with Fusobacterium nucleatum. It have an unusual requirement for N-acetylmuramic acid. So these are the studies which shows its pathogenicity. It is found in higher number in sites of destructive periodontal disease, active periodontal sites, or periodontal abscess than in gingivitis or health. It shows greater risk for alveolar bone loss, attachment loss, and tooth loss. It is shown to produce trypsin-like proteolytic activity and induce apoptotic cell death. Its levels were strongly related to increasing pocket depth. It was a major species which was found at site that converted from periodontal health to disease. Higher level at site which showed breakdown after therapy. It was decreased in frequency of detection and count after successful periodontal therapy. Listargen et al. Found that it most frequently detected in refractory subjects. Serum antibody elevated in periodontitis subject and extremely elevated in refractory subjects. Virulence factors, it produces several proteolytic enzymes, destroy immunoglobulins and factors of complement system. It induces apoptotic cell death. Trypsin-like proteases degrade smaller peptide. Spirochetes, these are gram-negative, anaerobic, helical-shaped, highly motile microorganism, has been implicated as the likely etiologic agent of NN. Ravitola intermedia. It is second black pigmented bacteroids to receive considerable interest. It is gram-negative, short, round-headed anaerobic rod have been shown to particularly elevate in anal periodontitis and progression sites. These are dark pigmented colonies. Its persistent after standard mechanical therapy has been shown to be associated with a large proportion of sites exhibit bleeding on probing. It has a high number of virulence properties exhibited and shown to reduce mixed infection on injection in laboratory animals. It invades oral epithelial cells. Fusobacterium nucleatum is a gram-negative anaerobic spindle-shaped rod. It is most common isolate which is found in cultural studies of subgingival plaque. Invasion of this species into human gingival cell in vitro was accompanied by an increased secretion of interleukin 8. It can induce apoptic cell death in mononuclear and PMN cells. Campylobacter rectus is a gram-negative anaerobic short motile vibrio. Unusual is that it utilizes hydrogen or formate as its energy source. It has formed small, convex, dry spreading or corroding colonies on blood agaptates. It is capable of stimulating human gingival fibroblasts to produce interleukin 6 and interleukin 8. Eikenella corridens is a gram-negative capnophilic asacrolytic regular small rod with blunt ends. It is most frequently and higher level in active sites and in sites which respond poorly to periodontal therapy. Has been also found in association with AAC in some lesions of localized juvenile periodontitis. It is shown to stimulate the production of matrix metalloproteinases, interleukin 6 and interleukin 8. Peptostreptococcus is a gram-positive anaerobic small asacrolytic coccus. It has two genotypes. Smooth genotype may frequently associated with periodontitis and rough is present more in health. Salmonella species are gram-negative curved rods, difficult to grow. Salmonella noxia was found at a higher proportion in chronic periodontitis subject. Eubacterium, they have been suggested due to their increase in diseased site. They are gram-positive, anaerobic, small, somewhat pleomorphic rods, elicited elevated antibody response in different forms of destructive periodontal disease. The Streptococci species, evidence suggests that Streptococci, Streptococcus angioinus, constellatus, and intermediates might contribute to disease progression in periodontal diseases. Other species like Enterobacterial flosia, pneumonia, Pseudomonas aeruginosa, Klebsiella are also seen in some of the cases. More recently, viruses like cytomegalo, Epstein-Barr, papilloma, and herpes simplex virus have been proposed to play a role in the etiology of periodontal disease, possibly by changing the host response to the local subgingival microbiota. Coming to microbiological diagnostic testing, so identification is done by direct field and phase contrast microscopy, but the disadvantage is individual organism cannot be identified, and advantage is the shift in periodontal microflora can be identified. Enzymatic assays, so plaque sample with any three combination of bacteria, paper strip is impregnated with BANA. BANA substrate breakdown and produces a blue-black color whose intensity is proportional to total amount of three microorganisms. Immune assays, it involves monoclonal or polyclonal antibodies against specific antigen. ELISA, immune fluorescence microscopy membrane assays comes under this. Advantages is low detection threshold, low cost and rapid and quantitative. Nucleic acid probes, it consists of nucleic acid sequencing. Probe sequence may be whole genomic, randomly cloned sequence of nucleic acid or synthetic oligonucleosides. Greater sensitivity than cultural method and viability of microorganism is not important. Polymerase chain reaction assays are also done. So these are the method to determine the microbial composition of oral samples. Thank you.